Hello, everyone. Welcome to today's live broadcast, phage antibody selection with Reichert SPR Systems. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Reichert Technologies Life Sciences. Reichert Technologies, part of Ametek, is a preeminent player in the design, manufacture, and sale and service of optical refractometers that are integral to the study and measurable effect of biological interactions. For more information, please visit www.reichertspr.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page to claim your credits. I would now like to introduce today's speaker, Dr. Mark Sullivan. Dr. Sullivan is a Research Assistant Professor, Department of Microbiology and Immunology, and Director, Biomolecular Interaction Laboratory at the University of Rochester. He received his Bachelor of Science in Chemistry from Purdue University and his PhD in Biochemistry from the University of Wisconsin-Madison. He was a postdoctoral fellow, Department of Microbiology and Immunology, University of Rochester, Rochester, New York. Research in Dr. Sullivan's laboratory is focused on the manipulation of immunoglobulin molecules to develop novel reagents for characterization of protein expression and structure. He and his colleagues at Reichert are using phage antibody technology in several areas of proteomics. I will now turn it over to Dr. Sullivan for his presentation.
Good morning. So this has been an interesting start. As you may know, Rochester just got hit with 5 inches of snow this morning, so welcome to spring. In any event, what I'd like to do today is give a brief outline of what we'll be covering. So I'll start with some basics of SPR technology, and then move on to a little bit of background on antibody generation and modification using phage display. Then I'll give some objectives and the rationale for investigating the Reichert system for phage display, and then show you some preliminary results we've obtained in fractionating phage on the SR7000 DC instrument. The main focus will be on binding and recovery of the phage, looking at the effect of single-chain valency, the display valency on binding profile, as well as actually getting some separations between positive binders and non-binders. So surface plasmon resonance has been around for just about the same amount of time as phage display. Both were developed around 1990. It's a label-free technique for analyzing biomolecular interactions in real time. It's an extremely precise mass sensor, and you can do quantitative binding measurements with it. It's a flow-based system, and the key components are the sensor chip, where the ligand is immobilized, a microfluidics system for delivering the binding partner, the analyte, and then finally an optical sensor to measure the changes in the resonance angle that is translated into a mass accumulation on the chip. And SPR really is the gold standard for determining kinetics of protein-protein interactions, and it's been used extensively in the field of the antibody technology, which is what I'll be addressing today. So just a couple key parts of the instrumentation that I want to go over. So the sensor chip is the small gold coated glass slide that's been surface functionalized for immobilization of ligands, and there are several options for doing that. Probably most commonly used are amine coupling with EDC and NHS. You can also put, using that same coupling system, you can put streptavidin or neutravidin down for binding biotinylated ligands. And finally, you could also use the thiols on the gold surface. And it's optionally can be formed with a polymer matrix to give a little bit of a balance between binding sites and signal strength. Although all the work I'll be describing today was done with the planar chip without the polymer on there. So antibodies form the largest class of new therapeutic agents. There's a large amount of activity in the pharmaceutical and biotech industry for deriving new antibodies for a variety of diseases and conditions. So the very first commercialized antibodies were mouse monoclonal antibodies that were humanized. Since then, and really concurrent with the development of the SPR technology, there are really now a variety of methods for generating human antibodies that are much faster than relying on hybridoma technology from mice. So the two methods that are most commonly used now, so the newest one really is the human antibodies derived from high throughput single B-cell cloning methods. This is very powerful, and many of the new anti-flu and anti-HIV antibodies have been developed using this methodology. However, a major limitation is that it's really limited to agents that humans have already been developed immunity to, and then that you can actually get access to their B cells for cloning. There's a large number of targets where that's not possible, and so more recent work has been devised as an alternative to generating antibodies, and that's basically the display technologies, of which there are several. Phage display is the oldest, was developed in late 80s, early 90s, and it is probably the most widely practiced and the simplest of the technologies to work with. Subsequently, there's been yeast display, ribosome mRNA display, as well as some cell-based methods. But phage display is really the workhorse of the display technologies for generating new antibodies or other proteins that act equivalently to antibodies. So this is just a graphic that sort of distinguishes the multiple display technologies. The key concept for all of these, of course, is that it links the biochemical properties of these displayed antibody to the genetic material that encodes the antibody, and the two travel together. And in a nutshell, that's what all the display technologies do. And I will only be talking about phage display, but there's much overlap in these technologies in terms of how they work and the kind of results they yield. So the main advantages for display technology in generating antibodies, of course, is that it is extraordinarily fast compared to the traditional hybridoma technologies. You can get antibodies, monoclonal binders in approximately a week with phage display and some of the other techniques. And that could frequently take months if you're relying on immunizations. Not only that, but we're not subject to limitations of immunogenicity, and so you can go after antibodies to self-proteins. And this is a very important advantage because much of the pharmaceutical industry is looking at antibodies that can mediate activity of various receptors and ion channels in humans, and so that you couldn't get this easily by immunizations because of the conserved nature of the targets. This technology will also avoid problems with immunodominance, and you have many different ways of presenting the target antigen to the various display systems, and so you have a lot of control on that. Another benefit is that at the end of the cycle, when you actually have obtained binders, you can easily get proteins for analysis. Of course, it doesn't use animals, which is useful. And you get, of course, the DNA encoding the antibody, which allows for further manipulations. Control of antigen presentation really is a fairly key advantage of the display technologies. And that allows you to use some custom conditions during enrichment. So, for example, to directly select for higher stability antibodies or control cross-reactivity of the selecting population as possible. So for the work that we've been doing, we're using a large, what's called a naive human phage antibody library. For most of these libraries that are available, the diversity ranges somewhere, for good libraries, between 10 to the 9th and 10 to the 11th clones. You can use, the antibodies can be expressed in phage display using either FABs or single chain FVs. All the work that I'll be talking about today is in the single chain FV format. Many of these libraries have been built using pooled cDNA from many donors to try to give maximum sequence diversity in the immunoglobulins. And the binding, the selective pressure that is applied in selecting and enriching phage antibody libraries is basically the shape complementarity between the target antigen and the antibodies. That lets us select antibodies to virtually any target, whether it's human, human antibody library on human targets is completely viable. And generally, the affinities that you get from these libraries range probably from 10 to 100 nanomolar from 10 to the 9th size naive libraries. I should mention that there are other methods of generating sequence diversity in libraries. So there's several approaches to making synthetic diversity on a single framework. They by and large give similar kinds of results as the naive libraries made from cDNA. However, many applications for therapeutics and diagnostics do require a higher affinity antibody, and oftentimes in the sub-nanomolar range. And that's unlikely to be generated from the kinds of libraries that most people might have access to. So as I said, from a 10 to the 9th size library, the 10 to 100 nanomolar is probably the upper bound for high affinity, unless one is extraordinarily lucky, of course. To generate higher affinity, what's typically done is you take candidate clones and then subject them to secondary diversification using either things like error-prone PCR, or also you can dope oligonucleotides and do defined positions where there's extra diversity created, and then reselect those second populations, secondary libraries, under a variety of methods to select for the very much higher affinity antibodies. Probably the most common method to do that uses biotinylated targets, and then using magnetic beads to select, enrich for clones with slower off rates. And there's a fair bit of procedural differences in how one does this. One of my favorites actually required week-long incubations in a beaker full of buffer to select for slow off rates. So just briefly on the library that I work with, the way this was generated was we use a collection of light and heavy chain family primers coupled with J primers to PCR the light chains and heavy chains from a pooled cDNA from quite a few donors, in the range in several hundred donors, really. And when you generate those two fragments, they can be overlapped using standard splice overlap PCR technology to assemble the single chain. In my case, almost all the single chains I work with are in the light to heavy orientation. There are some in the reverse orientation, which actually will become important later on. And then I used a vector that I built quite a few years ago when I was in industry, and it employs using the flag tag as an N-terminal epitope tag on the end of the amino terminal end of the single chain. And we use this mainly for detection, but as you'll see, it has some purification properties that also are attractive. In any event, all of the single chains have an N-terminal flag on them. And for those of you perhaps not familiar with what the flag is, it's an 8 amino acid sequence for which there are several very specific antibodies available for binding to it. And then the vector that I'm using is it's called AP36. It uses an alkaline phosphatase promoter to derive the single-chain gene 3 fusion protein to get the phage display. And the point that I want to make about this is that unlike a lot of other vectors that have been developed for phage display, the reason the Fo-A is attractive for a promoter is that it keeps the expression level rather low in the cell, and then when you're producing phage, it's a very low-level promoter in standard LB medium, and that encourages true monovalent display, or close as one can usually get to it. Some of the other vectors that are out there use repressed lac promoters, and in my opinion, those are much higher basal levels of expression. So the odds of getting multiple display on the antibodies rises significantly. And so in general, my experience has been you want to keep the expression level of the single chain gene 3 fusion as low as possible. So that's just all of the work that I'll be talking about is using this vector. And so this slide just reiterates how phage display actually works to get binders, at least in my view. It's actually just finding the right puzzle piece that binds to antigen in a complementary fashion so that you get a decent binding interaction that can be selected out by phage. There's probably some charge interactions buried into this as well, but by and large, what you're really doing is sifting a billion piece puzzle for the 4, 5, or 6 pieces that surround the surface, that are complementary to the surface of your target. So sort of the simple and easiest way to do phage enrichments are biopanning. This was originally described by George Smith in the late 80s. And it works well, and we use it all the time. So you take a microtiter well and passively coat target antigen in the bottom of it. And this is standard ELISA methodology. So you coat the target, you introduce your phage library to it, and let what can bind. You then go through a series of wash steps, and then you elute what remains in these wells, typically with acid or base, and then you get a population that comes off these wells, and then that is reintroduced back into E. coli, and you reinfect with helper phage to get a new secondary population or a second pool that then repeat the process. And after 2, 3, or 4 rounds of this, you then derive binders that you can identify by ELISA. So, so what drives these enrichments, of course, is just the difference in binding over background. And this is just a simple bar chart that I've used for many years. This was the very first single chain I ever cloned back in about 1991 when I was at Eastman Kodak. And it's an antibody against the hapten phenobarbital. And it's just illustrating that the difference in binding between putting this phage antibody on its target, being phenobarbital conjugated to BSA, relative to other irrelevant targets conjugated to BSA, or just in the case of this tacrolimus, directly bound to a plate, you can see at least 3 orders of magnitude increase in binding to target. So that's that 1,000-fold differential is what drives the enrichment of very large libraries, 10 to the 9th to 10 to the 10th diversity, down to a handful of clones after 2 or 3 rounds of enrichment. So the phage display cycle can be thought of in really 2 parts. So the first part that I just illustrated, and this is illustrated again in a little bit different way, but the same concept, is your library is applied to your target, you bind, you wash, you elute, and then those are re-amplified by a round of regrowth, and then you reapply that population, back around to the original, to another fresh round of binding and washing and elution. And that usually happens with most libraries, probably two to three rounds, sometimes 4 rounds. In my experience, it's often 2 rounds. It's obvious that it's working. Sometimes 3 rounds. Almost never have I used more than 3 rounds of enrichment for the library that I have, which is about 10 to the 9th diversity. But these things vary with libraries, I think, and vectors, and so your results may differ. Nevertheless, and this takes, at least the way I do it, it takes about 2 days a cycle to run the binding and regrow the eluted phage. So about 6 days, and you can do 3 rounds, and then you're plating and then amplifying the clones and you test confirming them by phage ELISA. And then often the initial clones you've got, they'll be moderate affinity, and you may need additional affinity enhancement, and that would be the secondary mutagenesis scheme, which basically is run conceptually very similar to this, except you're using methodologies, for example, with the soluble antigen and biotinylated target to actually drive for higher affinity. And you'll notice that in this slide, the mutagenesis was successful because mutagenesis is misspelled, so we know it worked. One of the other things that one might be doing sort of simultaneously in this cycle is taking several candidate clones and want to test the affinity directly, and that requires protein expression. And so when we look at this entire process, where we could try to use an instrument like the Reichert SPR instrument to try to accelerate and improve this process, there's really sort of 2 areas that we can think about. So the first is in the actual selection process. Can we use the microfluidics on the chip to possibly enhance the speed at which this happens? And so that's one. And then you can think also that it's possible that we could actually analyze individual clones in the phage level for binding rather than at the protein level. Now, not going to really cover this in too much detail, but we actually think this actually could be very powerful if we can overcome a few of the issues that I'll be describing momentarily. But beyond just accelerating this process, one possibility that comes to mind is to use the controls on the flow system and the buffers that we apply to try to improve the effectiveness of the process. So, for example, we could start to control the temperature, time, flow rate, concentration of eluents to try to specifically select for the kinds of antibodies we're looking for, and in which case that might make the whole process much more efficient. Now, this is also a possible way of accelerating this process. If we can eliminate some rounds of enrichment, potentially one could imagine doing a single round if one can eliminate the background and only get a highly enriched population of binders to survive after 1 round of enrichment. That would cut down the time considerably. And then the other possibility is to start to think about how to increase the selectivity of the antibodies simultaneously with the selection. So, for example, there are many cases where there are several targets that one is isolating an antibody to, maybe homologs, and you don't want to hit other versions of, say, a receptor, but you need some specificity for a particular isoform of a receptor. Then one way of doing this would be to do a selective subtractive enrichment, where you perhaps In a multi-channel instrument, you can use some of the lanes to select for binders to the ones you don't want, get rid of those, subtract them from a population that's then moved to a channel with the actual target. And in that way, we can force the selection to be isoform specific. And so that would be a very powerful method potentially if, for those targets where there are other isoforms present. Or the other possibility with the 4-channel instrument is to run up to 4 instruments simultaneously. And so that would allow you to multiplex efforts considerably. So one of the advantages that we saw in looking on the Reichert instrument for this is it's a relatively open system. And importantly, the fluidics are really quite simple and readily accessible. And so that means we can literally tear this thing apart, exchange the tubing. It's a very inexpensive components. The tubing is fairly cheap. The sensor slides are pretty cheap. There is no fluidics master control board that you have to purchase that is fairly pricey. And then we can easily collect the outflow from the instrument and reroute it so you can unhook the outflow and change that into a fraction collector, really. Or in our case, we actually did it manually and just simply collected tubes that can then be analyzed offline. So that, coupled with the flexibility to employ different elution buffers, flow rates, and temperatures, we think lends some real attractiveness to the idea of using this instrument to actually do the phage enrichments. And as I mentioned, you know, the appeal of doing a subtractive selection is rather strong also, because in my experience, some of the existing ways of doing this aren't really very effective. So the key questions we wanted to address in this project were really quite basic, but needed to be addressed before one could even think about some of these more exotic applications. So the first, you know, can antibody phage be applied, bind to a target, and then be recovered in the outflow in the system? We didn't really have any guarantees that this would actually work, and so we needed to demonstrate it. So another question was, if you do this and you do bind phage in the channel, do they generate a significant SPR signal that would be useful for monitoring not just enrichment, but also analyzing individual binders when you get them? This also was rather unknown because if you think about it, at least in phage display, you have a single antibody molecule, at least in monovalent display, attached to a multi-megadalton long filament. And the interaction between the single antibody and the target antigen has to be strong enough that the phage body itself gets held up and binds so that you can then recover it. And then in a flowing system of the SPR channel, it was not entirely clear, and at least on first principles, that this would work well. And then secondary to that is how many phage can bind to an immobilized target. That was also a concern, because I don't really know how to translate the amount of protein that you might immobilize to how many phage can bind there. And so the reason that number is rather important is because if you're doing library, either enrichment or secondary libraries where you're looking for affinity maturation, even though those libraries are probably smaller than the initial libraries one would use, the amount of phage that can bind to the target is going to be the upper bound of a useful library enrichment. So if you can only bind 1 million phages, then it didn't make any sense to make a library of more than 1 million members to try to fish something out, because it's almost guaranteed to miss what you're looking for. So so that was a question we needed to address. And then buried into all of this is, how does the valency of the displayed single chain affect the binding profile? Because while we attempt to make monovalent display antibody in using these monovalent vectors, they're unlikely to be all monovalent. There's undoubtedly some level of bivalency present. And if you have multiple antibodies on a single phage particle, those will come off the target much differently because they're binding by avidity effects. than monovalent would be. And so that could complicate, at least interpreting the data you're seeing. And then finally, what we really need to address is how to do the separation. I mean, we're basically going to be running the equivalent of a chromatography system. And the important parameters here are that, so normally when you bind, say, an antibody onto a chip, onto a target antigen on a chip, and switch it to the dissociation phase, you start dissociating antibody almost immediately, except for very, very high affinity ones. And so what does bind initially, when you switch over to the dissociation or wash step, if you will, things will start peeling off instantly. And we need to be able to sort of run the system long enough to get rid of the either things that don't bind because they're non-binders, actual binders who don't bind because they didn't bind in the time period that you floated across the target chip, or those have to sort of reduce in amount before we can then elute what is bound. And these are all going to be variable because different antibodies have different affinities. And so we needed to really sort out how this worked before we could even think about some of the more interesting applications down the road. So you might wonder, so how has SPR been used previously for binding analysis? Well, the answer is really not very much. As I said earlier, phage display and SPR were both developed in the early 90s. That's quite a while. There have been a few papers describing phage selection with SPR analysis, but the early papers didn't really give a whole lot of detail as to how this was done. And there's some more recent papers that have been published. A couple of years ago, there was a very interesting paper published looking at peptide display that they at least gave a pretty detailed analysis of what kind of SPR signal they were getting and how many phage they were putting on and the conditions under which they did that. But the phage, those are obligatorily multivalent display, which I'll go into a little bit in a few slides. And so that offers some challenges in interpreting what's happening on the SPR chip because of avidity and multi-site binding. But nevertheless, so we were hopeful though that if we can implement this control of the phage enrichment using these instrument fluidics on the Reichert, and if the SPR signal offers improved discrimination of affinity variants, that would be a major advance in the field. So we set out to do this by just taking sort of standard preparations of phage, which were generated after infection with a helper and in sort of the standard small-scale approaches that people do routinely, and used an antibody that I had previously selected to bind to BSA as a very inexpensive and relatively well-behaved target that we could work with and mobilize to good levels on the SPR chip. And we pretty much quickly discovered that using just a regular helper phage infected single chain prep, phage antibody prep, gave us some rather challenging problems. And that's because the titer of this is quite high, on the order of 10 to the 12th per mil in transducing units. And the phage is flowing through this, even though it's all a BSA binder, of course, you have a large amount of phage particles that have no single chain on it because of the nature of the monovalent display, where that's the idea, is that you want to keep the display level on your phage populations down below 1% or so. And those 99% nevertheless still look like your clone when you analyze them, and when you recover them in E. coli, you can't tell the difference. It's still a BSA single chain sitting in the vector. So what was happening was these things sort of, even though 99% go through very quickly on the first flow through fractions, there's an awful lot of tailing of the phage, and so that what was happening was it seemed to be obscuring detecting the bound ones that we eluted 15 or 20 minutes later. So that was, the non-displaying phage was a real problem with this, and so we had to come up with a workaround on that. And I might also add that at this point we also weren't really seeing any SPR signal, so that was problematic as well. So the solution that we employed to do this was to purify those phage that actually have single chain on the surface. And the way we can do that is, as I mentioned earlier, we have an N-terminal flag sequence on the end of the single chain. And so we can take a population of phage and first bind that phage, the high concentration phage population, to anti-flag antibody. And that's the most convenient way to doing this is simply coating an immunosorbed strip, 8-well strip with anti-flag, introducing the phage to it, let it bind. And one appealing advantage that we initially employed quite a bit was that we could take those phage and recover them without doing anything harsh to the bound phage. So rather than using low pH or high pH to elute that and run the risk that that could conceivably damage the antibodies, we actually can use 3X flag peptide and just incubate that to the washed strips. And that pops most of the anti-flag, the single chain bearing phage back off the plate and gets it in solution without any damage at all. And so that's actually what we tried to do. And so we could put that phage preparation, which I shorthand call M23X, and I'll put that into the system, and then monitor what comes through, and then what happens when you elute it. So just to diagram what this experiment is doing, is we're injecting fairly small amounts, mainly driven by time factor. We're injecting 100 microliters of these phage preps. We're using a fairly slow on rate, 10 microliters a minute. And the reason for that is, we've looked at some of the literature, those people that bothered to tell us this. And so the consensus seems to be a very slow flow rate for the binding phase of this is useful. And that sort of makes sense because, again, you're having to capture a multi-megadalton phage antibody with the interaction of a single antigen, target antibody complex. And so we were concerned that this would alter on rates dramatically relative to, say, an antibody or even an antibody fragment like an FAB or single chain. And so it seemed prudent to cut down the flow rate lower than we might otherwise do when analyzing, say, regular antibodies. So we flowed that on, and then after it's gone on, we would switch the flow rate back up to a more conventional. We typically use 50 microliters a minute, and run that for about 20 minutes to wash off the phage that are going to be, the non-binders or non-binding population, although technically they should all bind, but the reality is many come through initially, and so we have to wash that out before we try to elute with acid. And then we take these fractions and we can transduce them back into E. coli. So that measures how many phage in each fraction, as well as we can then just confirm what has happened in this experiment. And what we observed is, if the phage input's about 1 × 10⁸, we were getting in the eluate, which is, again, an acid treatment after about 20 minutes of washing. We've caught 2 fractions for eluate. We were getting about 3 × 10⁶, or 3% of the input. So that's a little lower than we see in a coated well. If we did this exact same experiment in a coated well, we'd probably get somewhere between 10 and 30% recovery. My suspicion is that this is mainly due to the difference between a static ELISA well and then a flowing system. So you might imagine that some of the phage, also because of the size, and they're not sitting there for a couple hours as would be typical in a coated well experiment, that we just haven't given them enough time to bind, and so you end up with a lower amount of phage that actually stick to the surface. So that's sort of the downside, is we have to balance giving it plenty of time to bind, but on the other hand, even bound phage should start bleeding off just by the normal off rates, especially in a flowing system like this. So the amount of phage you can actually recover upon elution is going to be, have to sort of be fine-tuned to take into account those 2 parameters. So here's the profile, the difference profile between the channels. And as you probably can see right away, you know, this is the on rate and the on phase, and we aren't seeing really any signal at all in the SPR. But nevertheless, we can pop these things off with the acid treatment here, and when we measured this, we actually did get this, and I'll show you some more data in a second. After we do this, we run an anti-BSA antibody just to verify that the surface is still functional, and this is I think at 26 microgram per mil antibody, and so you get a very nice robust signal here coming off of the antibody. So that's at least telling us that the surface was fine. So despite what doesn't look like a very impressive SPR signal, we nevertheless did get signal in the eluted phage, and so we were at least encouraged by this. Now, as I said, in our original experiments where we had the monovalent display, which is diagrammed here. So what happens is you get, at least in our hands with the helper and vector combination I'm using, we think that this maximum display is about 1% of the phage particles are displaying single chain, and so 99% don't. So this is what we were using to eliminate the 99%, so that we would hopefully not have to worry about those contaminating the outflow. But there's also a possibility of doing multivalent display, and the way we can do that is to use what's called hyperphage, which is a special helper that's deleted for gene 3, and we use that to infect the cells that contain the vector with the BSA antibody on it. All of the phage that are produced come out multivalent, and in general, it's believed that about 3 to 5 copies of the single chain or gene 3 protein are on the end of these phage particles. So the one downside to doing it this way is that with my vector, since the expression level is so low in general, the yield of phage is not great coming off hyperphage. So it's considerably lower than you would see with a standard helper. But nevertheless, it's plenty high to do these experiments. We usually get 10 to the 11th per mil with the hyperphage, and these are multivalent. So we wanted to use a multivalent display to answer this question. A, can we get a better signal? And B, how many phage bind to the surface in the channel where they're multivalent, and you presumably have a much higher rate of success of binding and staying on the chip, even when you switch to the wash. And in fact, we think that's exactly what we've seen. So now we take a preparation, in this case it was about 2 by 10 to the 11th phage per mil. We apply them to the, inject them in, and as you can see, we get a steady rise in the SPR signal during the on-rate phase. We switch to the wash phase, and you'll note that not a whole lot changes. It's pretty flat, and I think this is expected because, as I mentioned, this is multivalent, so the phage are likely bound by multiple binding interactions, and that won't come off easily. And so this is entirely consistent with that. So we do this experiment, and after 25 minutes, by monitoring the titer, we're actually getting down pretty low in the background, and so then we elute with acid, and we found that we could recover about 8 times 10 to the 7th phage, or about 25-fold higher than when we tried the monovalent approach. So the good news was, there is a signal that phage bind. We just have to figure out how to get it with the smaller number of phage in the monovalent display. But secondarily, there's plenty of capacity to bind, and so now we can appreciate that a much larger type libraries could be applied to a channel like this. I should mention that in this case, we put down about 1,500 micro-RIUs of BSA on the surface. So it is a fairly sizable amount of material that was bound. But nevertheless, we were gratified to see that we were getting a fair number of phage back. and certainly much larger number than we saw with the monovalent. So now that we were convinced that the key parts of this idea were at least viable, meaning that we could put phage on there, and we know the capacity is there. So we wanted to be able to ask really the basic question before anything else can be pursued is can we separate the binders and non-binders? So we used a rather artificial system. But the thing is that to simplify the expense and time involved in analyzing what happens in these enrichments, I came up with a trick that I could use to make it much simpler. And that is I had a different non-binding clone that it turned out was in the opposite orientation with respect to the heavy and light domains of the single chain. That I had used for some other experiments. And so the BSA binder is represented on the top, and that's my standard orientation, light chain, linker, heavy chain. And when you colony PCR transductants using primers that flank the single chain, actually the front primer flanks it up in the leader sequence, but the downstream primer that I used actually primes in the 3' end of the heavy chain domain, the VH. So it's a VHJ primer. So that PCR product would be about 800 bases. If you do the opposite orientation, then that primer is landing only about 400 bases away from the upstream primer, and so the PCR product you get using the same primers is only 400 bases. Now the advantage this has is that we could do simple colony PCR without doing any binding analysis to figure out which clone is represented in a particular colony by just running the PCR products on a sizing gel. And if it's 800, it's the BSA clone. If it's 400, it's the non-binding clone. So that's the experiment we set up. And so it's run very similar to the one I just showed previously. So we inject a mixture of phage. It's about a 1-to-1 mixture of the it was an anti-peptide antibody. That's the reverse orientation. And the peptide isn't the BSA, of course. And then then the BSA clone was mixed with it. And so that was set. We titered this. It was an M2, the M2 3X peptide purified. We set the input to about 50-50. Did the same kind of experiment. We inject for a minute, for 10 minutes at 10 microliters a minute, switched the wash speed back up to 50, collected fractions, and then did an acid solution at the end of the wash phase, which was, again, it was about 20 minutes. So then we get colonies from each fraction, so that gives us the titer of the fraction, so we can calculate how many phage are in each fraction. And then we subjected some of each fraction to colony PCR to measure the percent, the composition of the fraction in terms of percent between the negative peptide binder and then the positive BSA binder. So this is what a sample of those results look like. It's not, I didn't have everything on one gel, so there's a couple panels missing here. But nevertheless, what you're seeing is that you can easily distinguish between the BSA clone, which runs here, and then the peptide one, which runs at this size. And so it's very easy to see that we have a mixture, and then as you go through the wash phases, that mixture stays. You still get both things out there. I'm not showing on this particular gel wash 4, which I'd previously done, which again, it was a lower level, and it's starting to drop, and you're picking up more of you're still picking up the peptide one. But then when you switch to the eluate in this fraction and in this fraction, you see almost entirely, well, in these two samples, you see entirely the BSA binder coming off in the acid. In the other gels that I didn't include this, we actually picked up a single clone of the peptide binder in the eluate. in any event, and for those of you who are actually paying careful attention to this slide, you'll notice I actually have one clone here that gave both signals, and presumably that was a double colony that I picked inadvertently, and so both have both plasmids in the PCR reaction. In any event, though, it's a very clean experiment and very easy to see that we were getting selectively the BSA binder and the acid elution of this after the washes. So to summarize how this experiment worked, this is a plot of the amount of phage in each fraction. The width of these of the columns here is proportional to the volume of the fraction I collected. So the flow through is the smallest, and then the wash and the eluates were roughly 250 microliter fractions. And down here is I've tallied the BSA binder versus the non-binding peptide antibody in each fraction that we found. And as I said, in these two fractions, in the elution fractions, there was only a single clone out of the 28 that I analyzed by PCR. And so it looks like the basic system is functional in that we can get binders and we can flush the non-binders out and still recover a sizable number of the binders in the outflow. And so I think that validates our basic idea that we can use the SPR system to do enrichments, binding enrichments in the SPR instrument. And so that left us with some good feelings about this. So the key results, though, to summarize this, is that it's easy to apply the phage. We can fractionate them, and they're easy to recover from the outflow. It's, I did it manually, but you could easily imagine putting the outflow tube on like a Gilson fraction collector, the old style Gilson fraction collectors, and just collecting fractions. When we're putting on 10 to the 8th monovalent phage, we can get 1 to 5% off. That yield is probably dependent on both how much target is immobilized in the channel, but also on the affinity of the antibody that's binding to it. I could imagine that with different affinities, you'd get a somewhat different result on this, but we have yet to show this. The maximum binding is significantly higher, and then we validated that with the multivalent binder. So I don't think that we're going to be limited to doing, in terms of the amount of phage that can bind to a channel. Obviously, this is probably also a function of the amount of mobilized target. We haven't done a lot of experiments on checking how that affects the binding, but that, of course, would be a reasonable thing to do. Clearly, this would probably be much simpler if we could increase the number of displaying phage in the output without increasing the valency. So one possibility comes to mind is not using a static microtiter coated anti-flag setup to do this, but perhaps an actual small column of M2 agarose, anti-flag agarose, might work a little better. And then finally, of course, increasing the input phage might also be useful, although it's unclear to me how many phage bind in a coated well. So that's the same problem we had with the SPR channels. We don't really know that. So that's an area that needs to be worked on a bit to see what the best way to do this might be. We can clearly separate binders from non-binders, and that was what we were hoping to achieve. The parameters such as flow rate and elution buffer composition will likely require, again, some more customization. How that plays into the affinities of various antibodies is unclear. We didn't look at using. Some people like to use base to do the elutions. In my experience, the pH2 glycine has always worked better in terms of getting number of phage back off of a target, but other people may have other reasons for using base. I think there's a lot of other possibilities for composition of the elution or regeneration buffer, if you will, that might be worthwhile. There's a paper that was published not that long ago that developed a cocktail approach to optimizing regeneration conditions on SPR in a way that maximizes retention of structure of the target. Those buffers might be worth looking at in terms of doing the phage, because phage, basically, the phage elution is the same as, conceptually, and it's the same process that you would do a standard regeneration to do kinetic analysis by SPR. So, except that in the SPR, you want to retain the structure of your target, whereas in a phage system, we can dispense with that if we don't want to reuse the chip. So, in any event, there's probably some room there for optimization as well. Now, the SPR signal in the monovalent case, it doesn't really come into play if you're actually doing an enrichment for low abundance binders. You're not using the SPR signal. But having said that, I think it could be very useful if we can characterize individual clones with varying affinities by that, but it will be essential to do monovalent phage. And so the challenge will be, how easy and convenient is it to make a large amount of monovalent phage for a panel of candidates that you can then use and test to see how they look by SPR signal. That will require a little bit of work, but in fact, I'm thinking that actually could be really valuable in terms of accelerating the process of obtaining clones via phage display using the Reichert SPR for the actually enrichment mechanism. So the reason for that, of course, is that we'd be able to, we can skip the protein expression part of candidates. If we could do that and just do phage analysis, and people try to do that by competition ELISA, I guess would probably be the main route that most people would attempt to sift through their candidates to decide who's got maybe the highest affinity before moving that candidate on to an affinity enhancement program. How well that works is, I guess that depends. I mean, the times I've tried it, I haven't been altogether impressed with the discrimination in affinities. And so I think having a better system that you could do by using the SPR would be wonderful. But it's going to take some additional work to show that that's true, and mainly in terms of getting a signal on a monovalent. That's really the critical issue there. As we mentioned earlier, I think this has the potential for eliminating rounds of enrichment, especially if one does the pre-enrichment first with anti-flag and you're putting just binders on. I think that, coupled with some stringent wash conditions, might be able to cut down the number of cycles you need for 1 to 1 or 2. That would be a nice savings. And also, we might be able to use the elution conditions to actually select for higher affinity binders right off the bat. That would also be a useful trick to have. And finally, getting specificity to homologs or closely related proteins to your target would be really nice. And if we could actually run these enrichments directly on the chip, that would also be really nice. And I think that's entirely feasible. We haven't been in a position to try this yet, but in fact, I think that's something that could be very powerful, and would like to see that experiment done. And finally, if you're just doing different targets, you could put 4 targets on a 4-channel instrument. That also is a nice time saving as well. So again, just to summarize, where I think the basics, I think the basics look favorable. But there's still a lot of work to be done to really implement this in a system that you can rely on. And so, for example, one possible technique that we should probably test is soluble target elution. So you just, let's say, for example, we bound to BSA, then we just flow on BSA in the wash buffer, and that should help pick off as antibodies released from the surface, they can be bound up by the soluble target. So if you have running an affinity enrichment, we could probably just, look for things that survive introduction of various levels of the target in solution in the wash buffer. Also, manipulating flow rate and temperature could do the trick too. In particular, getting a nice little temperature bump as the wash buffer goes through might be useful in getting a higher affinity clones to be retained in the system. And again, as I mentioned, characterizing individual phage binding to rank order affinities would be a huge benefit if we can get the prep on the phage down to at least a manageable amount of work. And again, subtraction schemes I think would be highly desirable. So, and I just wanted to end with this promo for the PEGS meeting where Reichert will be have a booth, and be happy to field any questions if anyone has any. Thank you.
Thank you, Dr. Sullivan, for bringing that important information to us. Before we get to the questions, which we already have a few coming in, I just want to remind our audience to submit your questions through the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many of the questions as we can today. We do have our first question in the Q&A box. Why doesn't the phage binding and the multivalent binding reach a steady state?
Well, my sense of this is probably that we we may not have saturated the binding in the first place. At least, I mean, if you're talking about the multivalent one, it actually, we switched over to the, after we gave it an arbitrary amount of phage going in, it was stayed on, and I think in that case it's clear to me that the multivalency was preventing anything from coming off that channel. So that'd be my best explanation for that. It's just too much multivalent. And I should mention that there is a paper in the literature from the early 90s that was looking at a peptide by SPR, and they had they actually published the profile, and that thing was virtually not coming off as well. And unfortunately, they concluded that they had a low nanomolar binder. But I suspect that this was again, obligatorily multivalent, and I think that's actually what they were seeing, was not high affinity per se, but just a multivalent effect.
Okay, moving right on to our next question. Is the purity of phage preparation a key factor?
Well, I guess I'd have to qualify this answer by saying it's not, what do you mean by purity? I think it's helpful to do the anti-flag purification, because that definitely gets rid of virtually any impurities that you might have. But I have put more crude samples on here, and I guess the main problem, of course, is you don't want to clog the tubing. So, but I've put centrifuge preparations, and I think you could also filter phage and retain the phage go through like a 0.4 micron filter. So you can do that. So I don't think that level of purity is a problem. I mean, obviously you don't want to have a lot of bacteria or anything in this when you shove it through. And certainly you have to be really cautious about resuspending your phage if you're doing a PEG precipitation, which most do, because there's a lot of junk that doesn't redissolve when you do that. But I don't think it's necessarily a problem. And certainly you can run a 0.45 micron purification if one's concerned about it.
Great. Our next question, why did you choose to use the planar sensor chip as opposed to the polymeric coating?
That's an excellent question. We did this. This is primarily just my worry. So here's the thing. So SPR has been around for as long as phage display has. So that's, you know, since 1990. So, you know, that's a lot of time. And very few papers actually talk about putting phage on these chips. And I asked myself, why might that be? And okay, so one possibility was it doesn't work. Okay, well, I was hoping it wasn't that. But my suspicion was that the instruments that were basically available from many places are rather expensive instruments and have rather sophisticated and expensive to repair fluidics. And I'm thinking that people are concerned about putting on, you know, really those kind of preparations that someone else is going to be coming in with proteins that are, you know, a little more sensitive and concerned about it. So in thinking about this as well, is that the other possibility is that those are generally have the dextran surface on there. And as I said, you know, if you think about trying to drag along a several million Dalton phage and an antibody on the end of it, I was concerned about the kinetics of how that would happen. And if I add to those concerns the additional issue about penetrating the dextran layer, it seemed to me that that was possibly not going to work just on the basis of what's actually having to happen when it binds to a dextran layer where the targets could be buried down a little further. So my inclination was to use the planar to simplify this and not have to deal with the possible effects of the dextran. But I'll admit that I haven't done a lot of work with the dextran chips. It could be either fine. I just haven't tested it yet.
Okay, questions keep rolling in. Our next question, did you think about injecting a secondary antibody once the phage binds to increase the magnitude of the signal?
In fact, I did not think about doing that. That's probably a pretty good experiment, although it's hard to imagine that we'll decorate it enough to see a significant difference in mass. The other thing that one has to keep in mind is these phages are bound to the target by one end, and so most of the filament presumably is dangling up in the liquid phase quite a bit further from the surface. So it's sort of unclear to me, you know, how much bang for the buck you're going to get if in fact, since the SPR signal should be dropping off rather dramatically as it gets further away from the chip. One of the papers that I talked about, well, actually, I didn't talk about it, but I mentioned it, that was published using a fiber optic SPR signal. Actually has a diagram in their paper contrasting the two modes of binding by the gene 3 protein at one end of the phage versus gene 8 along the length of it, and how they were concerned about getting a differential signal based on how they bind. So it's possible, it is possible that you might increase it, and we should probably try this, but it's also possible that because of the end-on binding it, we wouldn't really see a whole lot of benefit to doing that. But I'll admit, it's a good experiment.
Great, thank you. We have time for just one last question before we wrap up. An attendee asks, do you think it's also working with peptide phage display?
Well, in principle, yes. In practice, doubtful. And the reason I say that is because as I understand this, and I've talked to one of the leading experts in peptide display about this, is that generally speaking, even from multivalent libraries, like they have libraries, for example, that you can get from New England Biolabs, generally speaking, the affinity of those peptides is going to be rather modest. And in that case, it's going to be hard to accumulate enough on the chip to spot that. Now, they are multivalent, so you do pick up the avidity effect. So it's possible, but so it might work for like an enrichment type experiment. But the paper I was just mentioning about the fiber optic system, they actually did everybody a favor. They actually they compared the SPR signal in their system, which admittedly I think is a custom system that isn't commercially available. So it's hard to interpret that. But they compared the ELISA signal to the SPR signal they see, and also quantitated how many phage they were putting in. And I can tell you that most of their phage clones that were in, well, in fact, all the phage clones that they reported that were in the 3 to 5 valent gene 3 system, you had to put in at least 2 by 10 to the 11th to get any signal whatsoever off of it, and they only got fairly modest signals when they went all the way up to 10 to the 12th phage. And that's actually kind of tedious to get on those phages. That's a lot of phage to put in. So, and by the way, if anyone wants to email us, I can provide that link to that paper. It's a pretty interesting paper. But anyway, my suspicion is it wouldn't work all that well for peptide phages. And I saw one of the other questions there that I want to go ahead and address this anyway, because I probably should have spent some time on it. Somebody asked a question about contamination in the system, and this, in fact, is a concern. You can use the sort of ultra clean protocols that are online that you can use, where you use acid, and you use base, you can use urea, you can use SDS. That helps. Swapping out the tubing is also helpful. But yes, in principle, if you've put a lot of a phage on and you want to do it again later on for the same target, that can be problematic. I do have a workaround on that for affinity maturation, but from basic libraries, if you're going to repeat an enrichment from a library like a naive library or a general library, you do have to be worried about it, and in fact that could be a little bit tricky to get it completely cleaned up, sterilized. But you can run these cleanup protocols, and they work pretty well. So it might be manageable.
Thank you, Dr. Sullivan. That is all the time we have for questions today. I'd just like to ask Dr. Sullivan if he has any final comments for our audience.
Well, you've been a great audience. No, seriously though, yeah, it's. I think I enjoy having to to be able to sort of look outside the box and have tried to do some non-conventional work on the Reichert SPR. And one thing I can tell you is that the Reichert system to me was much more flexible than maybe going to some of the other instruments, and so I think there's some potential there if we can just do a little bit more development work on it. But in any event, I appreciate the questions at the end. They're some very good questions.
Great. I'd like to thank Dr. Mark Sullivan again for bringing this information to us, and of course thank our sponsor, Reichert Technologies Life Sciences, for making this educational webinar possible. Attendees will be receiving an email from LabRoots to alert them when this broadcast will be available for replay. That replay will be available through October of 2016, and we would invite you to tell your colleagues who may have missed today's event about that replay. That's all we have for today. Thank you very much, and we hope we see you next time.